Right.
Hi, Takaaki.
It's 4 o'clock, so let's make a start. Everyone who's running a bit late is welcome to join us whenever they're ready. Welcome back to day number 2 of the CNS Comms collaboration between the Clinical Neuroscience Seminars and the journal Brain Structure and Function to present the articles from their special issue on structural connectivity of the cerebral cortex. And just as yesterday, we have the 2 editors of the special issue with us who will run the show today. You see me only for the questions. And we have the authors in the audience. So do ask your questions. For everyone over on YouTube, just write your questions in the chat and I'll bring it over here to ask the authors on your behalf. Now with that, over to you, Hiramasa.
So thank you very much for coming back. So this is the second day of structural connectivity of the cerebral cortex special issue symposium, which is jointly organized between CNS seminar and brain structure and function. So, well, this is the second day. Many people already attended the first day. I'm not going to repeat the same thing, but here's the schedule today. So I think the first talk is from Dr. Kaneko about occipital five-matter tract in the marmoset. Then we have a talk from Dr. Gambrielli about macaque parietal lobe. Then we have a talk from Dr. Andre about brain structure and internalizing externalizing behavior. Then we have a talk from Dr. Ionauchi about five-matter correlates of hemiface dominance in emotional expression. Then finally, we have a talk from Dr. Van der Weyden about application of the trajectory for glial tumor resection. Then at the end of the all talks, we have 15 minutes for a question, which are open to every talks. But at the end of each talk, we have 5 minutes time for question discussion with authors of each paper. So if you have a question, please use your microphone, or alternatively, you can also use the chat function to ask question.
Hello, I'm Takaaki Kaneko. Today, I'm going to talk about a paper published in Brain Structure and Function special issue. In this paper, we investigated white matter architecture in the marmoset, especially about the visual system by using the diffusion MRI. Marmoset is a small New World monkey which recently attracted attention in neuroscience community as a transgenic animal were potentially available in this primate species. Over the last several decades, one of the most influential models in primate cortical visual system is a two-stream hypothesis, which distinguishes dorsal visual streams involving with spatial information processing and ventral visual stream involving with object recognition. To date, we often see schematic diagram of visual system emphasizing a distinction between these two streams. However, classical as well as recent studies on the human brain reported the vertical occipital fascicles, VOF, which is a major white matter tract connecting dorsal and ventral occipital cortex. Whereas VOF has been already known in anatomical works performed in the late 19th century, it has been largely ignored in the neuroscience literature until recent diffusion MRI studies. The diffusion MRI study also demonstrates the existence of the VOF in macaques, suggesting a homology of this pathway between two species. The existence of the VOF suggests that two streams are not strictly independent, rather interact with each other. In this study, we investigated marmoset brains to further clarify key organization principles of VOF and neighboring white matter tracts. Marmoset is a New World monkey and evolutionarily more distant to humans as compared with macaque monkeys. One interesting feature of marmoset brain is that they lack complex xylification, unlike old world monkeys and humans. We hypothesized that studying in marmoset is not only useful for clarifying evolutionary principles, but also helps us to understand key organization principles because of their simplicity in sulcus folding. We analyzed diffusion MRI data collected from 2 ex vivo marmoset brain using 70 MRI scanner. The figure indicated a color-coded diffusion direction map in representative coronal or axial slices of the marmoset brain. Color indicates diffusion directions. Blue is superior, inferior, green is anterior, posterior, and red is left and right direction. The high-resolution marmoset diffusion data demonstrates that the occipital white matter of marmoset is highly organized as multiple sheet structure along lateral to medial axis. In the most medial level, we can see colossal fibers. Forceps major is connecting between left and right hemispheres. Lateral to the forceps major, the optic radiation, large green structure in this figure, is running anterior to posterior direction and connecting the LGN and the V1. Finally, we also found VOF-like structure, blue structure in this figure, is running superior to inferior direction and lateral to the optic radiation. Next, we reconstructed these major tracts using tractography and then compared with macaque and human. Here we found special organization principles seems to be preserved across species, such that forceps major is the most medial, optic radiation is lateral to that, and VOF is located most laterally. In marmoset, fiber tract connecting dorsal and ventral areas looks relatively large and prominent feature of the visual system. We termed this blue structure in the marmoset brain as BOF complex, since it extends to parietal and temporal cortex, which do not follow the definition of vertical occipital fascicles. We will come back to this point in later slides. We then estimated putative cortical endpoint of BOF complex in marmoset and compare it with previous macaque and human findings. Similar to macaque and human, marmoset BOF complex has endpoints in relatively earlier stage of dorsal and ventral streams, such as V3A or TO. However, we found one notable difference between marmoset and macaque. In marmoset, the endpoints of VOF complex extended anterior from occipital cortex. The dorsal endpoints were found around parietal areas, and the ventral endpoints observed inferior temporal areas. This analysis shows that the posterior portion of the marmoset VOF complex has a similar connectional motif to the VOF of macaque and human, but that the more anterior aspects more likely correspond to other tracts manifested as multiple different bundles in larger primates. In human, white matter tract connecting parietal and inferior temporal regions were termed as posterior arcuate fasciculus (PAF). Similar tracts has been reported in macaques. In human and macaques, PAF is separated from VOF by superior temporal sulcus. However, as we see marmoset diffusion results, this clear distinction is absent, since there is not apparent boundaries between anterior and posterior portion of VOF complex streamlines. These results suggest that while organization of BOF is quite common across 3 species. BOF complex is segregated into multiple fasciculus in large folded human and macaque brains. This is summary of our study. In this study, we compare the spatial organization of major occipital white matter tracts across primate species. We found that spatial organization of major occipital fasciculus, optic radiation, major forceps, particular occipital fasciculus were common across primates, suggesting that a key organization principle may be present at common anthropoid primate ancestor. However, the marmoset BOF complex is a continuous seed-like. However, the marmoset BOF complex is a continuous seed structure, including fibers which are categorized as distinct bundle in human and macaques. This difference may be attributed to the difference in brain size or relative cortical folding, whereby the same basic connectional pattern is fragmented into distinct fasciculi in larger, more xerified brains. Here I conclude my talk by tributes to Dr. Kartsis. In his seminal studies, he proposed quantitative method to delineate cerebral cortex using distribution of receptor. Here's an example by cholinergic muscarine M2 receptors, and you can see the sharp boundary at broadband area 41 and 42, or 1 and 3B here. Such observer-independent way to clarify white matter arc structure is currently missing, but as he has very innovative mind, in his very later career, he was seeking for the breakthrough in this direction. This is one of important contributions where he clarified the occipital white matter architecture of velvet monkey brain in detail, using state-of-the-art polarized light imaging. He made an important step in this line of study, so I believe in next years there must be growing literature on quantitative and objective delineation of white matter, and also further understanding of white matter architecture in various species. We will continue to be inspired by his insightful approach on neuroanatomy for next investigations of white matter architecture and evolution. Thank you for your attention.
Thank you very much for this talk. Are there questions in the audience? As yesterday, you can either raise your hand or just unmute your mic and ask directly.
I have a what should be a simple question, I think. Do you have any comment on Meyer's loop in the marmoset? So from the LGN, do the fibers go anterior before they go back, as in the human and I think in the macaque?
Takaki, can you unmute? Yeah.
For sure. Sorry, but could you repeat the question again?
Yeah, I'm sorry. So I'm taking you, I don't think you commented on Meyer's loop, and I would be interested in if you have a comment on that, whether it exists in the marmoset. In other words, the LGN going anterior before it goes back to the calcarine, etc.
Ah, I see. Not so much salient, but slightly it go to anterior first and then go to the calcarine circles. In that sense, it's common structure, I think.
I see. Thank you.
Lovely. Are there any other questions? Not that I can see. I received a question on another channel, and the question is that the vertical occipital fasciculus looks bigger in the marmoset. Did you look at the proportional size of the WAF in regards with the optic radiation in order to make cross-species comparisons? And this would be especially interesting for the role of the WAF posterior arcuate for reading.
That is a very interesting point. So, but we didn't perform the quantitative analysis. We didn't perform the quantification. But for visual, just a visual inspection, as you can see, the proportion of VOA complex is very large in marmoset. So, but as this is in marmoset, this VOA complex is not completely homologous to the VOF of human and macaque. And this is some kind of mixture of the different bundles in other species. So it's slightly difficult to talk about the functional specialization in VOF.
Okay, thank you. I don't see anyone else raising a hand at the moment. Again, if you can think of a question for later, we will have a general discussion afterwards. Back to you, Hiramasa.
Yes, thank you. Well, I think you have still have some question. Maybe I think feel free to use a chat function, or you can just give your question at the end of the whole session. But if there's no other question, maybe we can move on. Thank you, Takaaki. So the next talk is talk number 7 from Dr. Michela Gamberini. So the title of her talk is Structural Connectivity and Functional Properties of the Macaque Superior Parietal Lobe.
Welcome to everybody, and thank you for the invitation to this virtual symposium. Our review included in the special issue Structural Connectivity of the Cerebral Cortex aimed to study the structural and functional properties of areas located in the superior parietal lobe of the macaque, looking at possible homology between human and non-human primates in this part of the brain. This is a general overview of my talk today. I will analyze the structural subdivision of macaque superior parietal lobe, followed by the description of the cortical connections and the functional properties of macaque superior parietal lobe, ending with a brief comparison with the human organization of the superior parietal lobe and possible new perspective. So let's start with the structural subdivision of macaque superior parietal lobe. The focus of this work was the superior parietal lobe of the macaque. The parietal lobe of macaque monkey can be subdivided into an anterior and a posterior parts. The anterior part in yellow occupies the postcentral cortex, where the somatosensory primary area is located. The posterior part can be in turn subdivided into the superior parietal lobe and the inferior parietal lobe, separated by the intraparietal sulcus. According to Broadband, in 1909, the superior parietal lobe is almost entirely occupied by area 5. Starting from Fock and Fock in 1919, most studies in the last century claim that although the architectural pattern changed, moving rostrocaudally, the superior parietal lobe was entirely occupied by area 5. In agreement with this view, Pandya and Seltzer in 1982 proposed the subdivision of macaque superior parietal lobule in an area named PE anteriorly and another one named PC posteriorly, but both described as a variant of broadband area 5. At this level of the brain, the occipital and the parietal lobes meet together. And in the parietal lobe, many areas have been identified with different anatomical and functional properties. These areas are distributed along the exposed surface of the superior parietal lobe, the intraparietal sulcus, the parieto-occipital sulcus, and the precuneate cortex on the mesial surface of the hemisphere. Area B6 here shows a clear occipital psychoarchitectural pattern with a very thick layer 4. Differently, area B6A on top of area B6 show a typical parietal pattern with a larger and less dense layer 4. Although Brodmann did not examine the architectural pattern of cortex buried in the parieto-occipital sulcus, the parietal pattern of B6A strongly suggests that it represents an extension of Brodmann area 7 that from the exposed surface of the superior parietal lobe extends into the dorsal two-thirds of the anterior wall of the parieto-occipital sulcus. The mesial surface at the level of the superior parietal lobule, so the precuneate cortex, hosts the mesial sectors of area B6A, PEC, as well as area PGM and 31. Finally, in the lateral part of the superior parietal lobule, so the medial wall of the intraparietal sulcus is occupied by two areas, PAIP rostrally and MIP caudally, recognized on the basis of the male architecture. Now we take a look to the cortico-cortical connections of this area. Each area of the superior parietal lobule is connected to a set of parietal and premotor areas involved in specific and functional role. There is no time to go into detail, but it is evident, for example, for area PE, that this area is more connected with the frontal lobe and with somatosensory primary area. While area PC and B6A receive also connection from visual area and bimodal area located on the parietal lobe. Area PGM receive a strong connection from the mesial surface of the brain and the cingulate cortex, and area MIP is well connected with the frontal lobe and with the parietal cortex. In particular, I show few detail about the frontal connections of area V6A, PEC, and PE. All these areas are connected with the frontal lobe, but area V6A, represented in yellow, is more strongly connected with sector of premotor area F2, representing the upper limb. While area PC, represented in green, is more strongly connected with sector of motor and premotor cortex, represented the lower limb. PE, finally in red, receive input from motor and premotor cortical region, represented both the upper and the lower limb. These anatomical data are in agreement with the functional properties of this area. So let me show now the functional properties of this area. Few examples of that. We start with the somatosensory representation. From area B6A toward the area PE, there is a progressive increase of somatosensory modulation. Here is represented in a graphic form and in a more quantitative way the somatosensory representation in these three areas. The more evident difference is that area B6A, represented in yellow, has an exclusive representation of the arm, while PEC and PE are important representation of the lower limb together with the upper limb. In all cases, the main representation is related to the contralateral part of the body. Here you can see first the somatotopy of the primary somatosensory area, so the complex 3-1-2 of broadband areas. These areas have been studied in detail and show a complete representation of the body. Posteriorly to that is located the area PE, dominated by proprioceptive representation, and hosts a raw topographic map of the body with an over-representation of the arm and particularly of the hand. However, only the anterolateral part of area PE has been studied in detail. So the yellow, the light yellow region shown here. Whereas the medial part of this area is still quite unexplored. A recent paper of our group is focused exactly on that medial part of area PE. Therefore, it is possible that the lower limb are represented in this part of area PE, given also the fact that the leg is represented in the adjoint medial part of the somatosensory primary area. And finally, some example of area B6A. It is worth noting that the active arm movement are always more effective than passive movement in activating B6A neurons. Here are shown some examples of B6A neurons modulated by reaching and grasping arm movement, together with other functional properties useful in the control of arm movement, like the eye position, the object affordance, the spatial attention, and the saccadic eye movement. Overall, the functional properties of area B6A suggest that this medial posterior parietal area is involved in the analysis of vision for action and in the control of all phases of prehension during reach to grasp actions. Recently, our proposed subdivision of monkey superior parietal lobule has been confirmed with chemoarchitectonic technique. As you can see here, each area shows a specific architectural pattern. This work has been made in collaboration with Prof. Carl Zilles, and we will continue this fruitful collaboration with Dr. Nicola Palomero Gallagher in order to check the organization also of human SPL with the same methodology. We all thank the fundamental and tireless work of Professor Zilles to understand the organization of the primate brain. So, taking into account the anatomofunctional organization of monkey superior parietal lobule, the role of this part of the parietal lobe seems to be crucial in the control of arm action. With respect to the classic schema, in which the dorsomedial visual stream is dedicated to the control of reaching, and the dorsolateral visual stream is specifically for grasping, it has been demonstrated that the medial circuit actually is involved also in grasping control, and it has been recalled as reach-to-grasp network to underline the fact that is involved in all phases of prehension. Now we make a brief comparison with the human superior parietal lobule. After Broadband, author reported that macaque and human superior parietal lobe are different in the distribution of area 5, bordered in red, and area 7. We suggest that it is not the case. However, while in monkeys, area 5 extends in the lateral part of the brain, in human is confined more medially in the brain. It is plausible that the human area 5 was confined so medially because it has been pushed upward and medially by the growing of the associative inferior parietal cortex. Strong support to the view that human and non-human primates have a superior parietal lobule organized in a similar way also come from recent neuroimaging data. Cirino and co-workers have found in humans the presence of a second somatosensory homunculus, the parietal body area, PBA, in the superior parietal lobule. This is the classic location of Broadband area 5 in human, and the PBA is reminiscent of the raw topographical map of the body observed in macaque area P. Is activated by goal-directed limb movement, in particular reaching, but is not topographically organized, representing mainly leg and toe. So, with respect to what was known about the organization of the monkey superior parietal lobule, our view is different. Area V6 here is occipital cortex, only visual, so likely part of Broadband area 19. Area V6A is a parietal cortex, bimodal, so likely Broadband area 7. PC and MIP are again parietal cortex similar to V6A, so likely part of Broadband area 7. PE and PEIP are always parietal cortex, but are different from V6A because are only somatosensory, so likely Broadband area 5. According to our view, Broadband area 7, here in orange, is larger than previously supposed, including both V6A and PEC. And this new organization highlights a strong similarity between monkey and human superior parietal lobule. In the human, the dorsal Broadband area 7 has been recently cytoarchitectonically subdivided in 7A anteriorly and 7P posteriorly. That could be homologous to area PC and V6A in monkey. Finally, strong support to the view that human and non-human primates have superior parietal lobules organized in a similar way come also from recent neuroimaging data from the Pizzalis group, who described in human the counterpart of macaque area V6, V6A, PEC, and PE. So, on the basis of the similarity between the monkey and human superior parietal lobule, we can suggest this new perspective. It is possible to better understand the structure and function of human superior parietal lobule, starting from the evidence on monkey superior parietal lobule. This advancement of knowledge can improve rehabilitation procedure and ameliorate the neuronal control of prosthetic ends from the human health. I would thank the two grants, the European and the Italian grants that support this research, and I thank you all the research group at the University of Bologna in Italy, and I thank you all for your attention.
Lovely, thank you very much. Are there questions in the room?
I have a question. I think, yes, for example, in the case of B6, I think it's quite often appear in the context of optic flow. Then sometimes PEC also appears, then maybe B6A sometimes. Then I'm just wondering, do you have any thoughts about how much these kind of organization or maybe reaching selectivity, kind of movement selectivity can be potentially related to some optic flow selectivity, because maybe optic flow can be important key for guiding our action to the environment.
Thank you for your question. You are interested in B6A about modulation of optic flow. I well understood.
Yeah,
Okay.
well, yes, I think that your talk is mostly focusing on maybe mostly about reaching selectivity or maybe different effector. But I just wondering, in a different context, people are talking about these regions regarding how much the neurons or maybe fMRI can be responsive for optic force stimuli. I'm just wondering, are there some concrete theory to understand the organization of maybe B6, B6A, or PC in the context merging both the literature of the movement or some actual visual motion processing?
Okay, so I can say for sure that in all these areas from V6 to V6A to PEC also, there is a strong modulation by the direction of movement of stimuli. And we didn't make directly experiment related to optic flow, but both in human and non-human primates is known that there is a modulation for this kind of stimuli. And for example, for V6 is well known also for human in fMRI study, that V6 actually make a mapping of the motion in movement, of the visual stimulation that moving, and so could help in understanding the exact position of visual stimuli, even if the scene is moving. So it could help in create a map of the space around us in order to focus and to identify the spatial location of object in the peripersonal space.
Okay, maybe I would interpose and follow up rather briefly. In the context of optic flow, then let me bring up the anatomical result with Elena Bora. This was a paper in 2011, where we showed some connections from both PE and PG to the calcarine fissure. So this was to V1, peripheral vision, V1 and V2. It was not very dense, but they were consistent over several animals. And I would almost say the V2 was almost moderate, whereas V1 was sparse. So possibly, I don't know what your, if you have opinion or comment on that, but if you start talking about optic flow, you see why I might mention that.
Yeah, so in these areas, in particular V6 has a direct connection with V1 and with V2 also. So for sure, they receive information that could help in understanding that kind of stimuli and that reference point. And all these areas in the superior parietal lobe are as well connected each other. So V6 is well connected with V6A and V6A with PEC. So this flow of visual information is important to code the visual input that are from the scene in movement.
Anton, you raised your hand a while ago. I give the floor to you now.
Well, thank you very much. Nice talk. I also have a question about V6A. I know it as a visual area, but you basically depicted it as a more motor area. And you said that V6A is more responsive to passive limb movements than to active limb movements. Is that when the monkey is seeing the limbs, or when the eyes are closed?
So I'm not sure to be well understand the question, but in V6A we have both. So we have cells that are modulated by passive arm movement, and actually the majority are modulated by active arm movement, but there are both. And in case of active arm movement, the animal is, so the reaching and grasping task has been performed in a dark situation, so the animal is not able to see the arm moving toward the object or toward the target. So there is not the visual information during the reaching movement.
Great, I see Anton's happy with that answer. That's all we have time for, and we move on to the next talk. Thank you very much.
Thank you to all.
So thank you very much. So next is talk number 8. The speaker is Dr. Quinn Andre. The title of the talk is Brain Structure and Internalizing and Externalizing Behavior in Typically Developing Children and Adolescents.
Hi everyone, thanks for tuning in. My name is Quinn Andre, and today I will be presenting on brain structure and internalizing and externalizing behavior in typically developing children and adolescents. So to start off, I'll be giving an introduction to mental health and the cortical limbic system in children and adolescents. So what we know about mental health already is that by the age of 40, about half of people will have had or have a mental illness. Now, about half of those people will develop their mental illness by the age of 14, and about 2/3 by the age of 24. So what does this tell us? It tells us that this youth age group is critically important for the development of mental health. Now, in terms of brain development, we see that structural and functional brain abnormalities in the prefrontal cortex, or the PFC, and the limbic system, as well as the white matter regions, or white matter tracts that connect these regions, have been shown to be related to mental health disorders as well. But what is less well known about is the relationship between brain structure and mental health related to behaviors in typically developing children and adolescents. So that's what we wanted to look at. So our study aim was to describe the relationships between internalizing and externalizing behaviors and brain structure using structural and diffusion MRI methods in typically developing children and adolescents. To do so, we used 67 data sets from 48 typically developing participants. We had 26 males and 22 females. And so for our 67 data sets, we had 48 participants come in for a first visit and 19 return for a second visit. Our participants were between the ages of 6 and 16 years old, with a mean age of 11.3 years old. For imaging, we used a GE3 Tesla MRI, and we looked at T1 weighted images and diffusion tensor imaging, or DTI. And then for our T1 weighted images, we looked at specifically the amygdala, hippocampus, anterior cingulate cortex, the superior frontal gyrus, and the middle frontal gyrus. And then for our DTI, we looked at the fornix, cingulum, and uncinate fasciculus. And you can see these regions in our figure on the left-hand side. And then for DTI, the specific measures that we looked at were fractional anisotropy, or FA, and mean diffusivity, or MD. Now, for those of you who are not familiar with DTI, I'll just give you a little bit of direction to help you navigate some of the results. So for FA, with typical white matter development, we tend to see FA values increase throughout childhood and adolescence. And for MD values with typical white matter development, we tend to see those values decrease throughout childhood and adolescence. And then finally, for looking at mental health, we used the behavioral assessment system for children, and we looked at the parent rating scale, and we looked specifically at internalizing and externalizing scores. And basically, internalizing refers to internally directed behaviors, so that includes things like anxiety and depression. And externalizing refers to externally directed behaviors, so that includes things like conduct problems, aggression, hyperactivity, that kind of thing. Okay, so moving on to results now. First, I'll just take a moment to orient you to our figures. So on our x-axis, we have mean diffusivity of the bilateral cingulum. And then on our y-axis, we have internalizing scores. Now, for our internalizing scores, the higher scores refer to more concerning behaviors. So that would be things like more anxious behaviors, for example. And then for these figures in particular, we had a gender interaction. So we have split our cohort into males and females. So males are in blue and females are in red. Now you can see in both figures, males show a slight negative relationship between internalizing scores and MD of the bilateral cingulum. And females show a positive relationship such that higher internalizing scores or more concerning behaviors are related to higher MD values. Now, these gender differences could be interpreted a number of different ways. So these gender differences may be due to the earlier development in females, including that of the cingulum, or perhaps it's a relationship that is just unique to females, and this could highlight the increased prevalence of internalizing symptoms that we see in females that's across the lifespan. In addition, similarly to what we see in these figures, in previous case control studies with participants who've been diagnosed with mental health disorders, increased MD values were seen in white matter tracts when compared to controls. Okay, so for these figures, we are now looking at externalizing scores. Now, for the figure on the left, we have a negative relationship between externalizing scores and FA now, not MD. We're looking at fractional anisotropy. So FA of the left cingulum. Now, as I was explaining in the last slide, lower FA values in white matter tracts compared to controls is what we tend to see in those diagnosed with mental health disorders. But we're seeing similar trends here in a typically developing population with no prior diagnoses. So we're seeing those with higher externalizing scores are showing lower FA as well. Now on the right-hand side, we had an age interaction. So we have split our cohort by externalizing scores. And so those who have externalizing scores that are below and above the mean, and our scores are the raw externalizing scores. So below the mean is in purple and above the mean is in turquoise. Now, those that are below the mean show an age-FA relationship in the left uncinate, such that older children had higher FA values, and this is what we expect to see. Now, the group with higher externalizing scores didn't show this relationship. So finally, the main takeaways I would like for you to gain from this presentation are that the relationships that we've shown today highlight the importance of sub-threshold behaviors relevant to mental health, as these are associated with white matter alterations similar to those seen in mental health disorders, and they suggest microstructure of the uncinate and cingulum may be early indicators of susceptibility to mental health disorders. So I would just like to thank Dr. Catherine LaBelle and Dr. Bryce Gearhart and the entire LaBelle lab for everything they've done. They were absolutely wonderful to work with, as well for all their help on this project. I would also like to thank my funding sources for making this project possible. And then I would just like to take a moment to pay tribute to Professor Carl Ziles. And then, of course, I would like to thank you all for listening to my talk. I hope you enjoyed it.
Great, thank you. So if I'm not mistaken, I can't see Quinn amongst us. If I'm wrong, please quickly shout. But if I'm right, then the senior author of the paper, Catherine, kindly agreed to take the questions. So are there any questions in the audience? No one in here? Okay, I received a question over here, so let me read it out to you. Did you try to model your data using regression with all tracts rather than assessing tract mental health relationship in a 2x2 design?
A multivariate analysis, including the tracts together? No, we didn't look at that. Now I'm trying to go back and remember our rationale, but I think we were interested in the tracts separately, and we'd only chosen these this smaller selection of tracts given their involvement in mental health in other studies of disordered mental health. And so we were really interested in looking separately at the contributions of these tracts, but that could be an interesting thing to look at for sure. I would imagine we'd see a significant relationship given the number of relationships we saw here.
Lovely. Any other questions at the moment? Not that I can see. If I just overlooked you, please make yourself known. If not, you can ask the question later on as well in the general discussion. Thank you very much for taking those questions.
Yeah, no worries.
And we move on to the next talk.
Okay, so we can now move to the next talk. Next talk is talk number 9 from Dr. Stefano Ioannucci. The title of the talk is Why Matter Correlates of Hemiface Dominance in Happy and Sad Expression.
Hello everyone, my name is Stefano Ioannucci. Today I'm here to make a presentation for my publication on brain structure and function. First of all, let me start by paying homage to Dr. Carl Ziles. So I chose this citation by him, which says that afferent and intrinsic connections as well as cellular types and the properties are the structural basis of a brain region's function. I chose this citation because it stresses the importance of white matter for cognitive processing. While traditionally the focus has been on gray matter. And that is why for the work I'm presenting today, we instead focused on the white. And then today I am presenting the publication which is titled White Matter Correlates of Hemiphase Dominance in Happy and Sad Expression. So as the title says, the point of this work was to evaluate asymmetric emotional expression in healthy humans and to test relationships of this asymmetrical emotional expression with structural properties of the brain, specifically the connectivity, so the white matter of human brains. And to do so, we started from a very established paradigm which employs chimeric facial stimuli. So Sakai et al. In 1978 did this well-known publication where they took pictures of participants expressing several emotions, and then they used the pictures of these participants to create new stimuli that were made up of the left and right halves of the faces of the participants. And this is what we call a chimeric facial stimuli. So here you can see an example. In the center, there is a natural face expressing disgust in this case. And then to the left, there is a chimeric facial stimulus made with the left half of the face of the participant's face by mirroring the left half of the face. And on the right, we have a chimeric facial stimulus made by mirroring the right half of the face of this participant. So employing this stimuli, Sakai et al. Brought forth evidence that humans tend to express emotions more strongly on their left side of the face. So what they did was show a bunch of these pictures to other participants, and they were asked to rate the emotional expressivity of a single one of these stimuli, one at a time, many repetitions, on a Likert scale. Since then, this method has been established and used for the scientific investigation of asymmetry in the expression of emotions. So here you can see some examples. It's also been used on primates to see on which side of the face they express emotion more strongly, or also if they have a preference in the visual hemifield, to recognize emotions. So this is to say that facial chimeric stimuli have been extensively used in this line of research. However, they have been seldom used in direct comparison. So this is precisely what we did here. What we did was take pictures of our participants. Precisely, we took pictures of them in a happy condition or a sad condition. What we did was simply ask them either to look happy or to look sad without precise directions on which poses to assume with their facial muscles, as previously done. So contrarily to what was previously done. And then we made facial chimeric stimuli out of these pictures. Here you can see some examples. So here we have a left chimeric facial stimuli and a right chimeric facial stimuli for the happy condition and for the sad condition. And what we did was to put them like this in direct comparison and ask a second group of participants to rate the emotional expressivity of this stimuli. Precisely, they were asked to mark a line towards the stimulus that they evaluated as being more emotionally salient. Of course, they could place a mark at the center of the line, and that would mean there was no clear prevalence. So this is, we use these measures to derive an asymmetry index of emotional expressivity in our sample of 33 participants who were used as models for the pictures, and another sample of 20 participants were asked to evaluate this stimuli. What we found was actually, in this accordance with earlier accounts, was that there was no clear predominance of either side of the face, meaning that some people tended to express emotions more strongly on their left half of the face, while others tended to express them more strongly on the right half of the face, consistently across happy and sad conditions. But others yet did not show a clear predominance of either side of the face. We did find stronger results for happy, like clearer results for the happy condition than the sad condition. And this is something which is often found in the literature, because happy faces are more strongly differentiated from neutral faces with respect to sad faces. Also, if you ask someone to look happy, most people are going to pose in the same way, meaning that they're going to contract the muscles similarly and look happy in a similar way, more consistently than if you ask them to look sad, which is the expression of sadness, spontaneous expression of sadness is more variable in the general population. But as I mentioned, we did not only want to check if humans do express emotions more strongly on one side of the face or the other, but we also sought to evaluate the relation of this phenomenon with the brain structural properties. So what we did was base our a priori hypothesis on a set of white matter tracks, which are specifically the uncinate fasciculus, the SLF1, 2, and 3, and the cingulum, which to varying degree are involved, can be involved in the processing of emotions. And we derived the measures of these white matter tracks from our sample of 33 participants that we had used for the preparation of the chimeric stimuli. So first of all, concerning the lateralization of these white matter tracks in their brains, we reassuringly found that indeed these tracks are lateralized following certain general trends in the human population that were previously reported in the literature, meaning that we found the uncinate fasciculus to be rightwards lateralized. We found the SLF1, 2, and 3 to follow a ventral to dorsal right lateralization. And we found the cingulum to have a left lateralization, meaning that people consistently in our sample showed a bigger cingulum in their left hemisphere. Concerning the relationship of these white matter tracks with the behavioral asymmetry indexes of emotional asymmetric expression, we found the uncinate to be significantly related to the asymmetric expression of emotion, both in the happy and in the sad face. So what does this mean? It means that people who tended to express happiness or sadness, both, more strongly on one side of the face, were found to have a larger uncinate fasciculus in the corresponding hemisphere. So if a person had a consistently left side of the face more expressive in a happy and sad condition, they had a larger uncinate in their left hemisphere. Conversely, only for the happy condition, which I mentioned was the one where we had strongest result, we found the SLF1 to be contralaterally related, meaning that people who tended to show a more stronger smile, a smile that was rated as more emotionally expressive on the left side of the face, tended to have a larger SLF1 in the contralateral hemisphere, so in the right hemisphere. Although the SLF1 is not traditionally linked with emotions, some recent publications have found the SLF1 to be involved, the areas that the SLF1 targets in the supplementary motor area, to be involved in the production of orofacial movements, so smiling, lip licking, and this kind of movements that are strictly related to the phenomenon that we're looking at here. So concluding, what did we find? Well, first of all, we found that, can we claim that humans actually display emotions more strongly on one side of the face with respect to the other? Well, this method that we use that puts in direct comparison, the left chimeric stimuli with the right chimeric stimuli did not show a clear prevalence in the general population. And I think that it is a more apt instrument for this kind of scientific question. So we could not confirm this. We actually brought forward proof that there is not a clear predominance of the left face as was thought. And but what does it depend on? If someone does actually show the emotions more strongly on one side of the face to the other, what does it depend on? Well, we did find some clues that there are some white matter tracts that are significantly related to the asymmetric expression of emotion. And particularly for happy faces, it is the uncinate, ipsilaterally, and SLF1, contralaterally. And for sad expressions, we found that it was always this uncinate, ipsilaterally. And also we confirmed the previous anatomical claims on these tracts. So thank you for your attention.
Lovely. Are there any questions for Stefano, who's the first author, or also Michelle, who's the last author on this publication?
Well, if nobody
Yes,
else, maybe I can.
No,
yeah.
Valentina has a question.
Yeah, maybe, yeah, she should go.
Hi, thanks for your presentation. It's been really interesting. I was just wondering, have you assessed any control task, for example, on the ability of each participant to recognize emotions?
We didn't. Hi, thank you for your question. So we did not have a special control as in assessing the ability of participants to distinguish emotions. We just asked the participants to have no history of mental conditions that may impair their recognition of emotions. And we also adopted a series of strategies to control for pseudo neglect, for example, which is the tendency to, since it was a line bisection task, we presented half of the sample with the stimuli in a certain order or inverted. So we did pseudo randomizations on the presentations of the stimuli.
Okay, thank you. Yeah. Because I was just wondering whether one could extract like an index of one's own ability to recognize emotions and add it as a covariate in the analysis of, I don't know, regression or yes, correlation that you did on each tract. It would be yes, interesting to see whether this variable could affect anyhow each participant's assessment of his own emotions.
Yes, indeed. But we didn't think of this, which is indeed very interesting. We thought of contrasting the known biases that could be in such because there is a wealth of literature on the fact that there is a hemispheric dominance also in the prevalence of left or right hemifield. So we try to balance the presentation of the stimuli.
Okay, thank you.
Thank you, Stefano. I did cut you off earlier. Do you want to ask your question now?
Yeah, well, in the case of visual pathway, I think the left visual field is kind of right white matter, then right visual field goes to the left white matter. Then I'm not sure the similar rule can be applicable for the uncinate because it's not kind of connecting so-called visual areas. Then of course, maybe functional property of uncinate is not fully symmetric, but I think do you have any thoughts about what asymmetry of the uncinate means in terms of function?
Well, thank you for your question. And what we found was this rightwards lateralization of the uncinate. And we know that the right amygdala, which is connected by the right uncinate, is largely involved in the processing of emotions. And that may explain why the uncinate seems to play a role also in the type of process that we investigated here in the present work. I'm not sure if that answers your specific question.
Yeah, maybe kind of, but I think this is a task like maybe faces showing in the left or right, then person make a judgment like which one is emotionally more salient.
Yes.
Yeah, so in that case, maybe this kind of lateralization index is related to whether faces appear in the left or right.
Yes, but the uncinate was of the people, of the models.
Yeah.
Yeah.
Yeah, and we really balanced left and right. And the judge, we didn't look at the anatomy of the judge who made the judgment on the emotional expression. We used the average score to correlate.
I see. Thanks.
Lovely. Are there any other questions for Stefano? Not that I can see at the moment. Thank you, Stefano.
Okay.
That already brings us to the last talk for today. Over to you again.
Thanks. So time flies so quickly. So the next one is the last talk in the whole symposium. So talk number 10 from Dr. David Charles van der Weyen. So the title of the talk is The Role of Diffusion Tractography in Defining Glial Tumor Resection.
Hello, everyone. My name is David van der Weyen. I'm the first author of this review paper, The Role of Diffusion Tractography in Refining Glial Tumor Resection. Diffusion tractography is based on the movement of water molecules in the brain, and this is based on Brownian motion. Brownian motion is just molecules move in a random order when they are unrestrained. When they have some physical barriers, they will tend to move in a direction that's parallel to these barriers. Let's take an example here. We have a white matter tract here. And if we measure the diffusion here, it will be even in all direction, or we say isotropic. And here it will have a direction of diffusion, so we say it's anisotropic. And now let's get tensors, and tensors just indicate the direction of diffusion. And if we link those tensors together, we recreated a white matter tracts, which is what tractography really is. I'm going to talk a little bit about the difference between DTI and advanced tractography. DTI means diffusion tensor imaging, and it is kind of the first generation of diffusion tractography. It had a few limitations, namely crossing fibers, false negatives in peritumoral edema. And it doesn't get all the white matter as you know, the large white matter fascicle start to fan out, as you can see here. Newer tractography techniques, and there are many of them. This is not a paper that's trying to compare the different techniques. This is just a paper that's trying to see this technique in general, how can it help in brain surgery. So you can see a more advanced technique. They are much better at getting the white matter tracts when they start to fan out. This looks a lot like the sulcal and gyral pattern of the brain. This is another example showing the difference between the techniques. Here we have DTI. This is a temporal lobe tumor, and this is the arcuate fasciculus and the inferior longitudinal fasciculus. You can see DTI gets the core of the fiber pretty well, but more advanced the techniques also get a lot more detail on the white matter tracts. Let's talk about the effect of tumor on surrounding tissues. So tumor, when they grow, they distort the peritumoral white matter architecture. Low-grade tumor, they tend to displace peritumoral tissue without necessarily destroying them. High-grade tumor, they disrupt and they destroy the tissues that close by. Those regions were difficult for DTI. Classical DTI newer techniques are much better at it because tumors, when they grow, they create edema, which widens the gap between fibers, reduces axonal density, which reduces anisotropy and induces false negatives. This is just an example. Here in red we have the tumor, in orange we have the edema. You can see newer techniques, this is Hardy specifically, is a lot better at crossing than what DTI is capable of. Let's talk about the nature of glial disease, and for that I'm going to show you a little video. So those are glioblastoma cells, and you can see you have one cell here that's trying to get away. All right, it's almost there, and it's gone. Okay, so what this means is that glial cancer cells, it's a very infiltrative disease. So that makes it so that it's hard for the neurosurgeons to find a clear plane of resection that allows you to remove all the tumor and leave all the healthy brain tissue out. Even here on gadolinium enhanced MRI, you can see that the margin of the tumor is somewhat blurry, and you may be wondering, is there some healthy axons that move through this part? Tractography is very important here because it can be used as a tool to help you make decisions. Let's talk about the maximal safe resection principle. This principle means that the more you remove of the tumor, the better the clinical outcome. But this is only true as long as the patient's function and cognitive status is maintained. In other words, if you don't destroy some nearby healthy brain tissue. Because of that, the post-resection residual tumor tissue is more frequently located along the pathways critical for functional processing. Of course, if you're a neurosurgeon and you know there's some important white matter tracts close by to the tumor, when you resect the tumor close to those important fibers, you don't want to resect them too aggressively. Let's talk about the uses of tractography and how it can help us make those type of decisions. Where do we stop cutting? And where do we cut? One of the main uses of tractography is preoperative planning. So it allows you to visualize the macroscopic displacement and discontinuation in the white matter tracts surrounding the tumor, which allows you to plan the best surgical trajectory and have a sense of the structures to avoid during the resection. It's a tool to help judging the level of aggressiveness in resection. Research shows that it modified the surgical approach in about 14 to 21% of cases, which is pretty significant. This is an example here at our university. We have a pretty large frontal tumor. We were hesitating, do we tackle this tumor laterally or medially? We did DTI and we found out that there's a lot of white fibers from the singular cortex. Those fibers are very important for cognitive function. Therefore, we decided to take a medial approach in our resection of the tumor to spare those fibers. Another use of tractography, it can be paired with functional MRI. When you do that, you correlate structure and function. This paper was published in Brain Structure and Function, so I think it is very appropriate to have it here. And I'm pretty sure Carl Zels would agree. He said once, it's not enough to understand a single letter, it's not enough to understand a single word, you must understand sentences. And I think what he meant by that is when you study the brain, you have to look at it from different angles. And this is a pretty good way to do that. But talking about brain surgery specifically, when we pair the two, we can verify that the fibers we see are factual, and we can do that by calculating the percentage of fibers that reach its supposed destination. So if you think you have the arcuate fiber, and you can see that most of those fibers reach a part of the cortex that doesn't light up when the patient is talking, you may have another white matter tract. Things that can interfere with the fMRI ball signal: highly vascularized tumor and vascular disease. So this may be a limitation of this technique. You can use tractography and overlay it on the structural MRI that's used in neuronavigation. Neuronavigation is subject to brain shift, and we're going to talk about this a little bit later. So this shows you what neuronavigation is, if you weren't sure. So it's kind of a GPS map of the brain, and you have a pointer during surgery, and you point it to a place in the brain, and it correlates that location to the pre-operative scan. However, one limitation of this is brain shift. What is brain shift? The brain is a kind of jello, and it's enclosed in a hard shell, which is the skull. And when you do an opening in the skull, you are releasing pressure locally, and so you can kind of suck in the brain in that direction, especially if the brain has a mass, a lesion that exerts some mass effect in the brain. It will increase, the shift. Brain shift is unpredictable. It can be up to 25 millimeters, and it changes as surgery is going on. So you can kind of imagine how it could interfere with neuronavigations. The factors that affect it are gravity, the loss of cerebrospinal fluid, the per-operative removal of tissue, and the edema that's caused by the trauma of surgery. Hemocephalus can also play a role. Pretty graphic picture coming. So this is during a surgery that we did here. The patient had a lesion with mass effect, and when we opened the skull, the brain started to bulge out. We measured this bulging to be about 2 centimeters. This is tractography pre-op and post-op. There was a mass lesion here, and you can see that the location and even the shape a little bit of this white matter tract shifted during surgery. You can almost imagine it moving from here to here. How do you correct for brain shift? So you can use an ultrasound that's paired to the neuronavigational system, and you can check in real life how your white matter tract have shifted. You can, if you have an intraoperative MRI machine, which is probably not the case of most people, you can just rescan the person, and you can get an update on where the white matter tracts are. You can also use direct electrical stimulation, or DES, to validate the location of your white matter tract as seen on tractography. Let's talk about this a little bit. So DES is the use of electrodes to apply an electrical current to areas of the brain, temporarily disrupting their normal function. We can use the expected deficits to verify the accurate depiction of fibers by tractography. So we have two types. We have awake and asleep DES. Awake DES is under local anesthesia, and the patient is awake. The patient is asked to engage in a particular task while the neuron stimulates the area of the brain that is critical for that task. A classic task is language. We ask the patient count, name things, just talk, while we stimulate the arcuate fasciculus. And if the patient stops talking when we stimulate it, we know we're at the right place. So using this, we can check, is the tractography at the place where I think it is, and I check with the DES. Asleep DES, it has a more limited range of possibilities. It's mostly useful for evaluation of pure motor function. The concordance between DES and tractographical reconstruction range between 82% to 97% across studies. The use of tractography has been shown to decrease the time spent during DES and the number of electrical stimulations needed to locate the white matter bundles. So in other words, it reduces the time of surgery. Discrepancies between DES and tractography can be used to estimate the intraoperative brain shift, as we were saying earlier. The benefits of using tractography. In general, the use of tractography increased the total gross resection by 6 to 40%. Its effect on functional deterioration, this was a little harder to find. Few studies have a good control groups, but from what I could gather, it's 15 to 30% reduction in post-operative deficits, but I would take this number with a little grain of salt. Another benefits of tractography is that it's non-invasive in the way that it has no side effects, no radiation to the patient, except for the time spent in the MRI machine. Classical DTI sequences lasted less than 5 minutes. Nowadays, we can get some good tractography acquisition in about 5 to 10 minutes, so it's not too long. And with that, we can get around 60 directions of measure. How do we validate the location of tractography? How do we know that tractography really is compared to the real anatomy? So we have post-mortem studies. In those type of studies, we scan a dead piece of brain and then we dissect it and check, do the dissections match the tractographical tracts. This showed that advanced tractography was able to overcome DTI in multiple areas as fiber decussation, zones of triple crossings, high and complex angulations, and the terminal arborizations of fascicles. Correlation between histology and tractography shows a correlation superior to 0.9 in uniform fibers and superior to 0.8 in crossing fibers, with an average of 5 to 6 degrees in orientation errors. However, in regions of transition between white and gray matters, orientation error of as much as 90 degrees occurred. So this is a part of the brain where tractography has a hard time. One caveat though, ex vivo tractography can be constructed with maximal quality. And by that I mean there's no movement artifacts. We can use a very strong magnetic field that we would never use in clinics. We can also use a very long acquisition time. So we have a small voxel size and we have a high signal-to-noise ratio compared with clinical scans. Manganese enhanced MRI, so we inject manganese ions which are taken up inside the neurons by calcium channels and then actively transported to the synapse, where they are only released if an action potential triggers exocytosis. Then when the manganese ions are inside the synapse, they're taken up by the postsynaptic neurons and the same cycle begins anew. So we have a structural as well as a functional marker. The only downside of this is it's toxic, so we can't use it for humans, but it is very useful to check, to validate tractography. And research using this technique showed us that angular differences of less than 10 degrees with classical DTI and less than 7 degrees with advanced tractography. Another way of validating tractography is using phantom studies. So a phantom is a synthetic object with a known structure. It can act as ground truth. Tractography was found to be within 6 degrees of the true fiber orientation. So all of those methods of validations are in agreement with each other. What are the limitations of tractography? So although fiber crossing was a huge problem for earlier DTI techniques, newer algorithms are much better at dealing with it. We have new problems that are emerging though. Since the tractography algorithms now has more local directions to choose from, it is easier to make mistakes and reconstruct false anatomy. This gives us some false positives. Crossing fibers may corrupt the orientation estimations of the tensors, which causes the tractography algorithm to link together bundles that are distinct. One study estimated those false connections to be between 7 and 35%, so that's pretty high. Studies show that more than 90% of the bundles, the actual bundles, are actually depicted, which is good. However, quantitatively more false positives than true positives are in the reconstructions. So this highlights the current difficulty of the technique to control for false positives. And reinforces the view that tractographical data has to be interpreted by experienced eyes. If you know your anatomy well, you're probably going to be able to recognize the true fibers compared to the spurious ones. There are new approaches to tackle this problem that are being created right now, and we have computer vision that shows promise at recognizing bundle shapes with true anatomical significance, and this shows those new avenues seem promising. All right, let's summarize. So, diffusion tractography is a technique that has a great potential. It suffers, however, from a lack of standardization. What I mean by that is that there are a lot of different techniques that are being used by different labs around the world, and most of them are very good, However, they are rarely directly compared with one another, especially in clinical settings. So I think that's one thing that should be worked a little more in the future. Tractography in general was shown in the literature to lead to better outcomes if it's used properly. This is a technique that's highly user and technique dependent. And because of that, it's not yet ready for a generalized applications. I would not advise just any neurosurgeon to start using it right now. It needs experienced user to be effective. But however, we think that it has a bright future and that it will show great promise if it keeps to be improved. So that's the end. Thank you, everyone. I'm sorry this presentation was very rushed. I delved into many topics very briefly. There's a little bit more in the actual paper. I await your questions, and I hope that this makes people think, and that this may create some discussion in the field.
Lovely, thank you very much. Are there questions in the audience? I saw one in the chat already.
I have a question.
Oh, yeah, can hear someone. Someone have a question?
Hi, I have a question.
Yes.
So this is a great talk, and I appreciate your summarization of the current status. One question is, for example, at the beginning of this presentation, you showed how you visualize these fibers for the entire brain. And actually, the fibers are everywhere. So perhaps you have some set of fiber pathway that should be preserved during the surgery. Do you have any comment about which fibers should be preserved and which fibers can be resected?
Can you hear me?
Yes.
Okay. That's a very good question, and this is something that we are always thinking about. There's a lot of fibers in the brain that we are not 100% sure what they do. And there are some that we know that they are important. We usually call them eloquent fibers. The eloquent, there are the ones that we pay the most attention to. The ultimate goal would be to preserve all of them. As we were saying earlier with some of the spurious fibers, sometimes it's hard to know the fibers that we don't know very well. It's hard to know if they are true fibers or not. So we are always trying to preserve the ones that we know are important. And the other ones we try to cut as little as we can to achieve the maximal resection without damaging healthy tissue. But that's a good question, and we don't really have like the perfect answer yet. There's some people who say that all fibers should be considered eloquent, even if we're not sure exactly what they do. And after we do a surgery, we check, we do some tests, motor, cognitive tests, to see if the patient was affected. But again, we're only checking for the fibers that we know the function of. So there could be lots of deficits that are too subtle for us to measure. So are still a lot of gray areas.
Thank you.
I would be one of those people arguing that the fibers are important. And if we can't pick it up, then probably our assessments aren't subtle enough yet. But I don't think there's connections that we don't need.
Yeah. There's technically no neurons that you can say, okay, I can get that, no problem.
Exactly.
But life is sometimes you need to you need to go a little bit more aggressively, but the goal is to do as little as we can.
There was a question from there was a question from Leah.
Hi, thanks for your talk. I want to ask you if you think that a long tract analysis can help to assess the tumor infiltration and can give some insights about the microstructural integrity of the tracts. And also another question is that like with fMRI, when you usually like in my little experience, when we try to integrate the fMRI data in the neuronavigator system, every time we weren't sure about the threshold to use also to guide awake surgery and intra-surgery stimulation. So if you have question about also that the threshold used for fMRI.
So I've never myself used both fMRI and DTI. There's actually a lot. So this is a review of like what can be done, but I haven't done all of it. I presented on pairing fMRI and DTI because I thought it was a great idea. But I'm not sure about which threshold to use.
Okay. Yes, because like in my experience, like if they electrostimulated the white matter, like the arcuate fasciculus, like the speech arrest was something that happened really often. Instead, with fMRI, they weren't sure, so they always ask which threshold, if you have to include more activation or less activations.
Yeah.
And then this was a question. And also I want to ask you that in your literature revision, if a long tract analysis can help in this field.
Yeah, I think long tract analysis, so I'm not a professional on this technique either, but I think it would be helpful to control for the false positives, right? That was your question?
No, just wondering if it's something that is done in the literature. I saw some studies, if you think that could be like useful for a neurosurgeon point of view.
I think it's been done already a little bit, and I am pretty sure that it would be helpful because usually the false fibers are smaller, when you take, this long fiber approach, I think you can remove a lot of the spurious ones. And so when I read those studies that talked about the number of false positives, I was a little bit surprised because when I saw it myself, usually you can tell which are the significant fibers, and when we operate, we are not, like machines, just looking at the DTI tracts and then just cutting according to it. We kind of think about what's underneath and what are the bundles that we really want to preserve. So I think it's less of a problem as what strictly the number make it look like.
Okay, thank you.
Yes, Michelle has a question.
I think, it's not perfect, but we should put that back into its context. 15 years ago, when I was doing my PhD, I was doing it, at the Salpêtrière, and I had the chance to work with Hugues Dufault. But if you were to think that white matter was doing anything useful in the brain, people were thinking you were crazy. And hopefully 15 years later, I realized that it might be important, and we have a method to look at white matter tracks, and this is a progress, little step after little steps, and this is encouraging that now neurosurgeons look at tractography, but it's true that it is also limited in terms of its capacity to represent the true anatomy, and there are papers reporting accident that happen. People using tractography and boundaries for the surgery, and patients coming out of the surgery with an hemiplegia, for example. This is really step by step. I need to be used cautiously.
Yeah, and I think when you use other techniques to validate the location of your tract, I think you can be pretty safe with the resection. So as example, when we talked about earlier, by using electrical stimulation, you can check that the right is in the right spot. Brain shift, as we talked earlier, is one big limitation. But if you're careful, you can have the positive of tractography with taking precaution to avoid the negative.
Thank you very much. There is a question in the chat. Do you see any improvement with multi-shell DTI techniques for detecting false positive fibers in the clinical practice?
So I'm not exactly sure what multi-shell is. I have a colleague who's a tractographer who's here too. Guillaume, are you familiar with it?
Yeah, and I answer in the chat, but so yes, it could, multi-shell data could help because some techniques now could, because tractography is based on FODF, so some different directions in the brain, and now with multi-shell data and even with single shell data, but we could do multi-shell, multi-tissue FODF. So we have a certain type of FODF per tissue. And we could easily imagine that we could have a specialized FODF for the edema, for example, or the tumor, or the ring between the edema and the tumor core. So yes, it could help, but I don't know if it really helped or not. We never did this experiment. So yes, but I'm not sure.
Lovely, thank you. Can't see a question at the moment, so I would just chip in and ask, I guess, both of you a couple of questions. One is that you mentioned that DTI had its limitations, and you can see the core of the tract, but not necessarily all the streamlines. But if you use advanced methods, then you have kind of the opposite limitation, that you have a lot of reconstructions that may or may not be anatomically accurate. So when it comes to neurosurgery, I'm just wondering what your approach is as to what to trust and what actually guides your surgery in the end, given that the algorithm you choose changes how you anticipate the anatomy to look like, plus the presence of the tumor that also changes the anatomy that you expect from a healthy brain.
So this all comes down to clinical judgment. All of the surgeons who have used tractography in the operating room, they have operated without tractography for a long time. So they've been used to make those decisions without tractography, and they know when they've cut here or when they've had this type of tumors, they've had those deficits, and they look at the tract and they correlate that with their previous experience. So when we look at the tracts, we are like, okay, so this is this tract. I know this one is important. I know it's here, so I'm gonna try not to cut here. Sometimes when we reconstruct, we see things that we can tell that they're not true. So we have less hesitation about going in that direction. Sometimes we just don't know. And when we don't know, we usually do as if we did not have DTI, and we proceed as usual. So it's kind of it's not something that we use systemically, as I was saying earlier, like a machine. It's more something that guides us specifically when we have a few decisions to make. We don't look at all of the tracks that have been reconstructed.
And the second question I had was about the intraoperative tractography. People in the field who use tractography for anatomical studies, they usually spend a considerable amount of time doing the pre-processing and the processing of the data and validating their reconstructions. You don't have that time during surgery. So how reliable really do you think that ready to have tractography is intraoperatively?
So I think this question is more about intraoperative MRI. We don't have this at our university. I think there are very few in the world. The people who have done that, I'm not exactly sure how they tackled this problem, but they probably did they have a tractography pipeline probably, and they all did this pretty quickly during the surgery, because the longer you keep the skull open, the more you are open to infection. So it should be done pretty fast. But when we do it pre-surgery, every time it's like a case study. So it takes a little bit of more time and involvement and discussion. And the other ways to check it intraoperatively is with ultrasound. And this is just readjusting the white matter tracts. There's not like the whole processing that's involved. It's just kind of shifting the image within the brain.
Okay. I think Michelle had a question, and it's also time for a general discussion. So I'm happy to completely hand over to you, Michelle.
All right, let's move on to, Laszlo, go ahead.
So when we founded the journal with Carl in 2007, the idea was that we introduce a tool, a venue for structure function integration, unconstrained by method, level of analysis, species. It was a very fruitful venue. But I would like to ask you, because you all do at various level, various density, granularity. So of course, to translate structure to function is not automatic. So we can study as detailed as possible the circuitry. The function doesn't open up simple. We need to study also the semantics, the algorithm, how brain areas work. So I just would like to ask you, how do you see now, or in the future, fine function or structure constraint function? But how far we get closer to understand the brain? Of course, I really like the neurosurgeon approach. How far from Penfield we got using structural and functional tools. So this is indeed incredible. On the other hand, sometimes my sense was that to jump from structure to function, there is still a huge gap. So I just would like to open a more general discussion, how far we are closer now to understanding the brain.
Laszlo, I'll make one comment, and that is, as I'm sure we're all thinking about the same things. And one word that has come to my mind repeatedly is plasticity. So we talk all the time about how plastic the brain is, and it is. There's absolutely no question about it. And then we talk about fixed structure. So there's a gap there of sort. Certainly, there. I would agree with you, but I think there are answers that are coming, and one would be a whole brain imaging, I assume probably in animals. I was reviewing recently, so that there are many other people, I'm sure, but David Amaral has done combined lesions and then followed by imaging and showing differences in subsequent to amygdala lesions at different points. So I think I'm going to leave it at that, but I think one could expand a lot on plasticity and the positive and the negative of plasticity for brain structure function.
And also to make the link between the structure and the function, I think we're really missing large reviews that integrate the knowledge from axonal tracing with current new methods such as tractography. Regarding the plasticity, I always think about Marian Diamond work, and I think glial cells have like an essential contribution to plasticity in the brain. We have no neuroimaging approach in the clinical practice or even in research that I know that allow us to look at the activity or the contribution or the increase of glial cells in the brain. Did I just kill it like yesterday?
Maybe one comment from my side, if there's a free spot. I think we can link function and structural networks to some degree with certain approaches. One obvious approach is to combine functional connectivity and structural connectivity, and you can look at the convergence or the difference among these two. And functional connectivity is not always stable, it's state dependent. So to some degree it's state dependent, and you can look at which structural connections are relevant for certain functional states. So we can make a link between functional and structural connectivity. Another thing is research that I have done together with colleagues from neurosurgery. We combined deep brain stimulation with fiber tracking. So we looked at the connections of electrodes and what's the effect, the behavioral effect of stimulation of this electrode, and what are the connections of these electrodes. So that provides a link between function and structure. I think there are ways to go. However, it's still at the beginning that we are right now to do that.
Well, I think that one of the problems I have in general talking about this topic is, in many cases, the definition of the term function can be ambiguous, because if I'm just asking people like, what do you think about what the definition of function, it's kind of always ambiguous. In many cases, people don't have a clear answer. But in many cases, I think the difference between structure and function can be sometimes ambiguous, because Catherine talking about plasticity. So I think if we are just expanding time constant, I think the structure can be sometimes quite dynamic. But as if it looks like a little more dynamic system. But if we are talking about function in general, in many cases, we are also talking about millisecond scale, very fine dynamics causing some particular behavior, but particular psychological processing. Of course, there are some gradual difference between function and the structure, but I'm not sure how much there's a very clear gap between them. But I think getting back to the point, I think it's quite important to think about, if we are talking about what the structure function relationship, what the exact meaning of the function, I think it's quite much study dependent, context dependent. I think it can be a little complicated.
Well, yeah, and the very way we define like function and different function is completely biased by observation. So the way we understood the different function and behavior the human body and the human mind can do is completely related to the observation of disorders. And then a little bit of imagination with functional MRI, but I think we're really far from having a crisp, balanced depiction of the functional making of the human brain.
So I would probably say the same thing, just repeating myself. In a way, we're still at the first stages of neuroscience, of brain science, of localization of function versus distributed. And that's something that hasn't really gone away. We still need to, I would agree with Hiramasa, we still need to re-examine that and press the facts, press the reality, and press the nomenclature, probably.
And then there is a lot of subjectivity in the way we observe things. Like you change your algorithm in tractography, you change completely the way you look at connections. You change the method, you change the way you see connections. We've seen like those wonderful connections with like very, very strange trajectories that Kathleen has been showing us yesterday. And the same goes for functional connectivity. You had regressors, use different algorithms, you might have slightly different networks. So it looks to me like, you know, it's going to be a hard way to merge everything together into a comprehensive vision if we don't want to model noise, which would be, in that case, our subjectivity.
Could I add an observation about that Michael said, is that sometimes we try to understand the functional meaning of one specific connections, looking first to the most strong connection that we can observe. But actually sometimes the more interesting and tricky, funny things are related to the low connection. So sometimes there are also few connection put interesting indication, interesting notes about the functional meaning of two areas that are directly connected.
Yeah, and let me, go ahead.
I would totally agree with that we are slightly biased in terms of what we're looking at, partially because some of the connections are just easier to dissect because we know the anatomy and our methods allow us to study them, whereas other connections may be too small for our resolution at the time, especially if we're talking clinical data, for example.
Yeah.
But also, again, coming with the clinical background, when you look at behavior, for example, you don't always have the time in the clinic to do a full comprehensive assessment that gives you tiny facets and flavors of different disorders. Sometimes you do when you do the research study, but again, our tests they're wonderful, but for some facets of our behavior, they're just not good enough yet. So there's still a lot of room for new assessments and new methods in terms of the granularity of what we can look at.
And in terms of moving from the localizationism to the white matter, I feel like so there's been some progress. Before we were putting function to brain regions. Now we put function on white matter tracks, step by step. But maybe we should move on like to statistical models that look at, you know, an integrative way where you put like all the tracks in the same model and you see how they interact together in the achievement of the behavior or the function that you look at, because it is, you know, it is as simple as thinking that a single region will be a function or a single track will do a function. It is probably more the contribution of a large network of regions, and we have models for that, and we can extract them with tractography. It's just a matter of like putting them together in the same model.
Just a side note for those of you who don't monitor the chat, there is a side discussion going on with a lot of references. So you might want to just grab those references for your information.
Sorry, I evoked that discussion about parietal cortex because I think that Michaela mentioned that maybe parietal cortex have been forgotten too, then people just go through for this section. But when we are just writing some editorial for this special sequence, I think the one topic I discussed with Michelle is, well, I think that talking about structural connectivity, I think looking at many neuroscience textbook or computational model, I think the models are quite schematic or sometimes very simplistic, like maybe looking at many model like, well, this one is connected to where, this one is connected to where. It's quite binary in many cases, or sometimes it's little weighted. But I think reality, I think, as we've seen the many talks in the over 2 days, I think the connectivity itself is quite diverse because in the single axon level, I think there are lots of other collateral, there are lots of diversity in the type of connection, or maybe synaptic connection can be also quite diverse, or even if you are looking at more macroscopic level, I think the fiber tracks have some individual differences, maybe related to some function. Then I think there's a question is how much we need to think about this diversity of the structural connectivity. I think that many of neuroscience think it is kind of noise. Well, this is connected or not. I think a simpler model is much more easily appreciated. But in reality, I think the structural connectivity can be more complicated concepts. And I think we need some appropriately complex model to understand what's the principle. Because if the model is too complicated, maybe it's not interpretable or we are modeling the noise. But I think there should be appropriate level of sophistication of the way to understand structural connectivity in order to really link to function.
Where do we draw the bar, you know, where it is complex enough, but not too complex that we completely lose track of like how it is working? Where would you put, you know, this bar?
That's a complicated question because I think the practical limitation is the level of information we can get from each study, each measurement method, or each individual modality. I think that sets the bar practically for individual studies. But in a real perspective, I think different methods are picking up different aspects. Then if we are combining many different literatures, there's a rich information, but there's always a challenge to interpret. Reading a paper about the method which I'm not personally familiar with, that requires some communication among different disciplines in order to really make a right model. Because individual paper, I think we can rely on appropriate level of complexity we can get from each data. But for whole perspective, I think we need to talk more.
And that's exactly why we're doing those conferences after the special issue
to
integrate knowledge and confront positions. All right, is there any additional comment or discussion that you would bring to the floor? All right, I don't see any hand rising, and it is about time to finish or conclude. So it's been really wonderful. I had a great time listening to the talk and discussing with all of you, and I hope we'll have the chance to do that again in the future. This is the last CNS event of the year, but you can still come on Monday morning if you want to come to our Neurochino, where we discuss the exciting science of last week, drinking a coffee all together. It is 9:30 a.m. Paris time. Sorry for those of you who are in the United States, but you can still catch up and watch the Neurochino replay on the YouTube channel. And leave comments if you have questions about it. I'd like to thank Laszlo for trusting in us, organizing this event and leading this special issue with Hiromasa. It's been really great. I'd like to thank you all for your participation and writing the papers. So I think that was like really, really good special issue. I'd like to thank the Neuro Network, of course, and well, I hope to see you all again in a future event and probably a future special issue. There is one of Hiromasa as a visual system at the moment that is open, And I have one currently on brain asymmetries if you're interested. So if you have contribution, just drop us a little mail. With this one, I thank you all very much, and I wish you a very good weekend if you're in Europe, a very good night if you're in Japan, and a very good day if you're in the United States.
Thank you. Bye.